You know, our DNA is kind of like a living document. It's constantly being written on, scarred by everything from sunlight to the chemicals we run into. And these events, they leave behind tiny clues, little fingerprints that can tell us what might cause diseases like cancer. But finding those clues, it's like trying to solve a crime with contaminated evidence. A new tool, though, is completely changing the game.
Okay, let's dive right into what that challenge looks like. On the left, you've got the observed profile. It's just a messy mix of all kinds of signals. It really is like a crime scene with 1,000 different fingerprints, but we're only looking for one culprit. The whole goal is to pull out that clean exposure signature you see on the right from all that background noise in the middle.
So, what are these clues? Well, scientists have a name for them: mutational signatures. You can really think of them as unique fingerprints, left behind by all the biological processes and environmental stuff that have shaped our genomes over a lifetime. And finding these things? Oh, it's a huge deal. It's absolutely crucial for discovering new biomarkers and for really truly understanding the mechanics of how cancer even develops. But there's a catch. A really big one that has plagued researchers for years. The problem? The real kicker is that these vital fingerprints, these critical clues, are often just buried. They're buried in a massive, overwhelming amount of noise. And this noise, it comes from literally everywhere. It comes from our own body's natural aging processes, from little artifacts that pop up during lab work, and even from the sequencing machines themselves. Trying to isolate one specific signature in all of this, it's like trying to hear a single whisper in the middle of a hurricane. So you'd think we'd have tools for this, right? Well, we do, but they all really struggle with this noise problem. A simple subtraction method can actually create results that are biologically impossible. Other tools like NMF, that's non-negative matrix factorization, they need huge sample sizes, which just isn't practical for most experiments. And then there are others, like NNLS, that can end up overfitting the data. It became super clear that we urgently needed a better, more robust way to do this. And this is where SIG-RescuR comes into the picture. It's a new computational framework that was developed to solve this exact problem. It's designed, just like the name says, to rescue the true signal from all that noise. Now this is where it gets really cool. Check out how SIG-RescuR actually thinks. It doesn't just blindly subtract the background noise. No, instead, it uses this powerful statistical approach called Bayesian inference, and we'll unpack that in a second, to intelligently model and separate the real exposure signal from all that confounding background stuff. So, what is this Bayesian inference thing? Well, in simple terms, think of it like a super smart detective. It uses what it already knows, plus any new evidence, to weigh all the possibilities and find the most probable culprit. And this part is crucial. It also tells us how confident it is in its answer. It gives us a whole range of possibilities, not just a single all-or-nothing guess. Okay, so the big question is, how does SIG-RescuR actually stack up against the other methods in a head-to-head comparison? Well, let me tell you, the results are pretty staggering. And wow, this first chart just says it all. We're talking about accuracy here, how well each tool reconstructs the original number of mutations. Just look at SIG-RescuR, right there at 0. It's nearly perfect. Meanwhile, the other tools are all over the place, either adding or losing hundreds of mutations. That's a major, major flaw. And it's not just about getting the numbers right. It's also the best at finding the true signal without making a bunch of false calls. On a plot like this, you want to be in the top right corner. That means you have high precision and high sensitivity. And look who's sitting right there all by itself, SIG-RescuR, outperforming all the other methods. And get this, it's not a one-trick pony. Not at all. This thing is incredibly versatile. It works across all the major types of mutations, from single letter changes, which are called single base substitutions, to insertions and deletions of DNA, and even more complex changes. It's also fully compatible with the very latest, highest precision sequencing technologies out there. Okay, so all this technical power is impressive, for sure, but what does it actually mean for the real world? Why does this matter for cancer research and, for patients? It means that scientists can now find signals that were completely invisible before. Take this study, for example. In patients treated with 5-FU, a very common chemotherapy drug, SIG-RescuR found the drug's specific fingerprint, that signature SBS17B, in 8 different samples where it was totally undetectable before. Just look at the huge jump in the cleaned bars. It's incredible. So, what this really is, it's a total game changer. It's a transformative framework that helps us more reliably identify things that cause cancer and get clearer, more trustworthy results from even the most advanced genetic technologies we have. So let's just recap the key advantages here, because they're huge. It preserves the integrity of the original data. It works even with a tiny number of samples. It gives us that crucial, credible interval to show how certain it is. And it just strengthens results across the board.
You know, this breakthrough in separating signal from noise isn't just some small step forward. It really opens up a whole new level of discovery in cancer genomics and toxicology. The ability to finally hear that whisper so clearly, well, it poses a pretty exciting question for the future, doesn't it?